Hello, everyone. Good day. Here we are in the second part of our discussion on the family Neisseria and related organisms. And in this lecture, our focus would be on Neisseria meningitidis and other Neisseria species and those organisms that can be mistakenly identified as members of the genus Neisseria. So to start with, take note that even if Neisseria meningitidis is a primary pathogen, yet it can be found as a commensal in the upper tract. Specifically, the organism is carried asymptomatically in 5 to 10 or even up to 30% of the population as part of the normal flora in the pharynx and oropharynx, particularly among adolescents and young adults, especially those military recruits. Take note that humans are the only natural hosts of Neisseria meningitidis, and that transmission of infection occurs primarily as a result of the acquisition of infected respiratory droplets expelled from a carrier that is in close contact with a susceptible host. So let's say, for example, in this illustration, we have here a respiratory droplet that carries the microbe that has been expelled from a carrier, and once a susceptible host is in close proximity, this can be inhaled by the susceptible host, and the organism within the respiratory droplet could establish infection in the nasopharynx. So take note that even though the microbe is transferred through respiratory secretions, but it is not airborne, and therefore should require close contacts between the carrier and the susceptible host. And it is for this reason that outbreaks caused by Neisseria meningitidis usually occur in crowded living places, such as in student dormitories, and as I said earlier, in military camps. The primary diseases that are caused by Neisseria meningitidis are meningitis and meningococcemia. When we say meningitis, it refers to the inflammation of the meninges of the brain and the fluid that surrounds the brain and the spinal cord. And meningococcemia refers to the presence of Neisseria meningitidis in the blood, resulting to septicemia. And once the organism is in the blood, it will have the ability to disseminate to other parts of the body, causing invasive diseases. However, apart from causing meningitis and meningococcemia, Neisseria meningitidis had been isolated from urogenital and rectal sites due to unsafe oral anal sex, or even oral and genital sex. There are many risk factors that may put an individual at higher risk of acquiring Neisseria meningitidis infection. First on the list are those asplenic individuals. These are individuals that lack spleens, or individuals that do not have functional spleens, or those individuals whose spleens have been surgically removed. So take note that if a spleen is not functioning well, or the absence of spleen indicates that there is also absence or the lack of splenic macrophages, which are important in filtering and phagocytosing bacteria that are introduced via the bloodstream. And those individuals that are asplenic virtually have no splenic macrophages. Also, take note that the spleen plays a vital role in the immune response of encapsulated bacteria. And take note that Neisseria meningitidis is an encapsulated bacteria. Also, persons with complement deficiencies, particularly those with deficient C5, C6, C7, and C8, are also at higher risk of acquiring Neisseria meningitidis. Take note that these complement components that participate in the late phase of complement activation are critical in supporting complement-dependent bactericidal activity against Neisseria species, and the lack or deficiency of these complement components put individuals at higher risk of acquiring Neisseria meningitidis. Other risk factors include active and passive smoking, as it weakens the respiratory tract cells that line in there, making them more susceptible to Neisseria meningitidis colonization, and also those individuals living in crowded conditions. Although Neisseria gonorrhoeae and Neisseria meningitidis share virtually the same virulence factors as discussed earlier, the main virulence factor of Neisseria meningitidis is its polysaccharide capsule, as you can see here. So in this illustration, it shows that the cell wall of Neisseria meningitidis has an extra layer, which is composed of polysaccharides, and this is the capsule of the bacterium. And it is the one that renders Neisseria meningitidis resistant to phagocytosis. The capsules of Neisseria meningitidis species vary antigenetically, and because of these antigenic variations, there are 13 serogroups identified. We have serogroups A, B, C, D, H, I, K, L, X, Y, Z, 29E, and W135. In the US, the predominant serotypes or serogroups that are associated with Neisseria meningitidis infection is or are those that are under the serotype or serogroup BCY, while infections that occur worldwide are associated with serotype or serogroups ABCY and W135. The incubation period of Neisseria meningitidis infection ranges between 1 to 10 days. And when a susceptible host acquires a respiratory droplet that carries the microbe that is coming from a carrier, the organism, as it enters the respiratory tract, primarily colonize the nasopharyngeal mucosa. And as it replicates in there, it gains access to the bloodstream. And once in the blood, Neisseria meningitidis can cause any of these two primary diseases. We have fulminant meningococcemia and meningitis. Fulminant meningococcemia refers to the presence of Neisseria meningitidis in the blood, resulting to sepsis. And the presence of rapidly growing Neisseria meningitidis would also result to the release of lipooligosaccharide, particularly the lipid A component, which contains the endotoxin, as discussed earlier, simply because the rapidly growing Neisseria species would form outer membrane blebs that carry the LOS. And once endotoxins are present in large amounts, which is what we call endotoxemia, these endotoxins would trigger macrophages to secrete large amounts of pro-inflammatory cytokines that can heighten the inflammatory response, and this could be manifested by purpura and petechial rash, which indicate continuous hemorrhages or leakage in the capillaries. Tachycardia, which is characterized by heartbeats over 100 beats per minute, simply because of hypotension. When there is hypotension, the heart beats faster to attempt to increase blood pressure to deliver oxygen to other parts of the body. But over time, when there is continued leakage of blood in the capillaries, there would not be enough oxygen that can be delivered to other organs of the body, particularly the brain and the kidneys, and this would result to shock. And because there is prolonged inflammation, there is prolonged capillary leakage, there would be thrombosis, which is also known as disseminated intravascular coagulation. And when fulminant meningococcemia is not treated, it would result to death of the infected patient. Another type of disease that can be caused when Neisseria meningitidis enters the blood and subsequently breaches the blood-brain barrier is meningitis, characterized by an abrupt onset of frontal headache, neck stiffness, confusion, and photophobia. We have here Waterhouse-Friderichsen syndrome, and take note that this is a complication of meningococcemia. So when Neisseria meningitidis reaches the bloodstream, it further disseminates to other parts of the body, including the adrenal gland, and it causes hemorrhages of the adrenal gland. And when adrenal glands are affected, they fail to function normally. Take note that Waterhouse-Friderichsen syndrome, or WFS, occurs in pediatric population, especially at infancy or childhood, and it is always considered a pediatric emergency simply because prognosis is poor, meaning to say that if a patient with meningococcemia already suffers Waterhouse-Friderichsen syndrome, then it is difficult to reverse even with the administration of treatment. And most of the time, it will result to the death of the infant or the child. With regard to laboratory diagnosis, the primary specimens that can be collected are, of course, CSF for the diagnosis of meningitis, and blood specimens for the diagnosis of meningococcemia. And take note that just like Neisseria gonorrhoeae, the presence of SPS in blood culture system could inhibit Neisseria meningitidis, and therefore it requires also the addition of gelatin to hydrolyze, or to neutralize rather, to neutralize SPS. Nasopharyngeal swabs can be collected to determine the carrier status of an asymptomatic individual. And for disseminated infections, aspirates from joint fluids can be collected. And for those with urogenital infections due to unsafe oral genital sexual behaviors, then sputum and samples from urogenital sites could also be collected. And once specimens are received, especially those that are not obtained from anatomical sites that are populated with flora, then a direct microscopy is needed, and we can expect to see intracellular and extracellular gram-negative diplococci, as you can see here, similar to the expected findings when you do direct microscopy from specimens collected for Neisseria gonorrhoeae. And take note that these findings, although indicates the presence of Neisseria species, but it's always just presumptive because many bacteria share the same morphology and arrangement, and many bacteria also can have intracellular existence to polymorphonuclear leukocytes. And this time, after this, we can inoculate the specimen suspected of having Neisseria gonorrhoeae primarily on blood agar plate. But as I said earlier, we can also utilize enriched chocolate agar plate with inhibitors such as modified Thayer-Martin or Martin-Lewis if specimens were collected from anatomical sites containing normal microbiota. And once inoculated, primary culture media should be incubated in the same atmospheric conditions described for Neisseria gonorrhoeae, which is at 35 to 37 degrees Celsius at 3 to 10% CO2 with increased humidity. However, this time, colonies must be observed after incubation. And if there are no colonies that are formed after 24 hours to 48 hours of incubation, you can extend incubation up to 72 hours. And when colonies are formed, you can observe for the presence of round, smooth, glistening, and gray colonies, as you can see in this picture here. And take note of the mucoid consistency of the colony simply because Neisseria meningitidis contain a polysaccharide capsule on its surface. And that polysaccharide capsule gives its mucoid colony appearance. And once colonies are grown, you can proceed to indirect microscopy, and you have to have seen gram-negative diplococci with adjacent ends, as these are the presumptive findings that would warrant us or you to perform oxidase test as the presumptive test. And after performing oxidase test, either through a filter paper disc or by directly dropping the oxidase reagent to a colony that exhibited gram-negative diplococci with adjacent ends, then you can proceed to its definitive tool of identification, which is carbohydrate utilization using CPA medium. So going back to oxidase reaction, if the colony that exhibited gram-negative diplococci with adjacent ends tested positive for oxidase test, then it is indicative for the presence of Neisseria species, but we cannot point out yet because there are many Neisseria species under the genus. So we have to perform the definitive test. So in this test, as I discussed earlier, this is a carbohydrate fermentation test that utilizes phenol red as the indicator, and each tube here contains 1% of glucose, maltose, lactose, and sucrose. I repeat, these tubes here contain 1% glucose, maltose, lactose, and sucrose. And we are going to observe at which sugar or sugars the isolated bacterium is able to exhibit fermentation. Now, take note that for Neisseria meningitidis, the organism ferments glucose and maltose, thereby causing a yellow coloration in the CTA media that contain 1% glucose and 1% maltose. And in this way, we can now isolate and speciate Neisseria meningitidis. And because we are sure that it is Neisseria meningitidis, then we can proceed to antimicrobial susceptibility testing. Okay, let's have now the summary for the algorithm for the laboratory identification of Neisseria species. So as I said, let's begin with specimen accessioning and evaluation. And once deemed accepted, you proceed to direct microscopy and perform gram staining and observe for the presence of intracellular or extracellular gram-negative diplococci, which are coffee bean or bean-shaped in appearance. And of course, inoculate the specimen on its appropriate primary isolation media. For Neisseria gonorrhoeae, you are supposed to inoculate it on an enriched chocolate agar plate with inhibitors and antibiotics. While for Neisseria meningitidis, the standard culture medium should be sheep blood agar, but can be inoculated on enriched chocolate agar plate if specimens were collected from anatomical sites that are populated with flora. And after that, you may perform colony morphology to examine for colony characteristics, and of course, indirect microscopy through gram staining, and you should be able to observe the presence of gram-negative diplococci that are bean-shaped in appearance for you to be able to proceed to the presumptive oxidase test, that if tests positive, then it would indicate that the genus present is Neisseria, but if negative, then you should consider other organisms. So if the oxidase test show a positive result, then you have to proceed to the definitive tool of identification, which is carbohydrate utilization test using CTA, to which glucose fermentation occurs in Neisseria gonorrhoeae, and maltose and glucose degradation occurs with Neisseria meningitis. And apart from the conventional CTA method, you can also utilize multi-test or chromogenic substrate method to determine carbohydrate utilization. And once isolated, you can perform AST, you can even perform beta-lactamase testing, and of course, reporting of results. Next, we have here a summary of the non-pathogenic Neisseria. Although they are non-pathogenic, but they are of clinical importance since these non-pathogenic ones closely resemble pathogenic Neisseria species and must be differentiated in order for us to perfectly identify the causative agent and of course for physicians to come up with appropriate treatment and management plan. So first we have here Neisseria cinerea. It resembles pathogenic Neisseria gonorrhoeae, and these are the commonalities. Take note that both of these organisms, when tested for glucose utilization in commercial systems, such as using multi-test and chromogenic substrate methods, they may have similar glucose utilization reaction, and both bacteria show similarities with T3 colonies. And to differentiate them, take note that Neisseria cinerea grows on sheep blood agar, while N. gonorrhoeae cannot because it's fastidious. And N. cinerea is susceptible to colistin. N. flavescens and N. cinerea, although both are non-pathogenic, but can be similar to each other in terms of their asaccharolithic reactions when tested for carbohydrate utilization using the conventional CTA medium. So to differentiate the two, take note that N. flavescens grows on sheep blood agar or chocolate agar plate at 22 degrees with yellow colonies because of the pigment that it produces. Neisseria lactamica and Neisseria meningitidis are similar in terms of their ability to ferment glucose and maltose in carbohydrate utilization assay. Although Neisseria lactamica can be a fermenter of lactose but may produce a delayed reaction. That is why in 24 hours of incubation, Neisseria lactamica and Neisseria meningitidis would appear similar in terms of glucose and maltose utilization because lactose fermentation occurs later after subsequent incubation because of its delayed process of fermentation. So to differentiate Neisseria lactamica, which is a late lactose fermenter, from N. meningitidis, which is a non-lactose fermenter, then you can perform ONPG test. And take note, Neisseria lactamica tests positive for ONPG because it is a late lactose fermenting Neisseria species, while Neisseria meningitidis will test negative for ONPG because it is a non-lactose fermenting Neisseria species. So in this way, we can differentiate the two. Then we have Neisseria mucosa, which resembles Neisseria sicca and Neisseria subflava biovar perflava. They have similar reactions in carbohydrate utilization pattern. However, take note that Neisseria mucosa is differentiated from Neisseria sicca and Neisseria subflava biovar perflava simply because it has the capacity to reduce nitrite further to nitrogen gas. And then lastly, we have Neisseria polysaccharia, that resembles Neisseria meningitidis in terms of glucose and maltose utilization. However, it is different from Neisseria meningitidis because of its ability to grow on nutrient agar and produce polysaccharides from 1% or 5% sucrose. We are down to the last part of our lesson, and in this portion, we are going to talk about the family Moraxellaceae, which is one of the families of bacteria that can be mistakenly identified as member of the family Neisseriaceae. So the three families that are, or the three genera rather, that are part of the family Moraxellaceae are the following. We have the genus Moraxella, the genus Acinetobacter, and the genus Psychrobacter. So take note that under the genus Moraxella, Moraxella catarrhalis is the only species isolated in humans and a part of the commensal of the upper tract. Moraxella catarrhalis is an opportunistic pathogen. Generally, it is harmless but may cause infections to individuals that are immunosuppressed, especially those that are in the AIDS stage of HIV or those receiving immunosuppressive therapy or those that are undergoing chemotherapeutic agents for treatment of cancer. However, in some healthy adults and children, this opportunistic pathogen may cause upper respiratory tract infections and may cause lower respiratory tract infections among persons with chronic obstructive pulmonary disease. Moreover, Moraxella catarrhalis is the third most common cause of acute otitis media, which is an inflammation of the inner ear, especially affecting the eardrum, and sinusitis in children. And for immunosuppressed individuals, this organism may cause life-threatening systemic diseases when it is able to spread through the blood and to other tissues of the body. So microscopically, using direct microscopy, we can observe the presence of intracellular gram-negative diplococci, especially from respiratory specimens, and this would indicate possible infections caused by this organism. So this is what I have been talking about earlier, that if you're able to observe the presence of intracellular gram-negative diplococci, although it may suggest the presence of Neisseria, but it is just simply presumptive as many organisms, especially Moraxella catarrhalis, can also be found in respiratory tract specimens having the same morphology and arrangement that of Neisseria species, which is gram-negative diplococci and intracellular. So therefore, finding of this appearance in the microscope, specifically finding of gram-negative diplococci that are intracellular, may suggest Neisseria and may suggest Moraxella. And therefore, they must be differentiated. And on top of that, it is also similar with some strains of Neisseria gonorrhoeae because of its ability to produce the enzyme beta-lactamase. For the specimen collection, for patients suffering from otitis media, samples can be collected from middle ear effusions, or basically the fluid that builds up in the middle ear during otitis media infection. We can also collect specimens from the nasopharynx sinus aspirates and sputum aspirates, including bronchial aspirates. On culture, when grown on blood agar plate or chocolate agar plate, the bacterium shows smooth. As you can see here, it shows smooth, opaque gray to white colonies. And the colonies are described as hockey puck. Why it is described as hockey puck? Because when the colonies are pushed across the plate with a loop, the colonies are movable, but even if it's movable, it remains intact. When colonies have been subsequently incubated and becomes old, they may appear to have a wagon wheel appearance. So here is a wagon wheel as a reference, and this is how older colonies of Moraxella catarrhalis would look like when they are incubated for a prolonged period of time on culture media. And take note that this bacteria tolerate lower temperature and grows well at 28 degrees Celsius, and they can be inhibited by colistin present in the media, although take note, resistant strains may grow. So in terms of similarities with Neisseria, take note that apart from being gram-negative diplococci that exist intracellularly in polymorphonuclear leukocytes, Moraxella catarrhalis also are oxidase positive and catalase positive. And take note, we learned earlier that Neisseria species are oxidase positive and catalase positive. However, one way to differentiate Moraxella catarrhalis from Neisseria species, particularly those from pathogenic Neisseria, which are Neisseria gonorrhoeae and Neisseria meningitidis, is their asaccharyllytic reactions when they are incubated in a CTA medium containing the carbohydrates. Meaning to say, because they are asaccharyllytic, they are unable to utilize any of the carbohydrates present in the medium aerobically or anaerobically when they are inoculated in a CTA medium. On top of that, apart from their inability to produce acidic byproducts in the presence of carbohydrates when incubated using the CTA medium, their ability to grow on nutrient agar, their ability to produce DNAs, enzyme, and their ability to produce a positive reaction in the butyrase esterase reaction test using tributylene as the substrate are also important differential characteristics that differentiate Moraxella from Neisseria. So at this point, let's talk about DNase test. So DNase by Moraxella catarrhalis, as it is the one that produces DNase. So if the organism is inoculated on a DNase medium, it cleaves the DNA methyl green complex, and it would result to the appearance of a faded color green in the medium. So why would it fade if DNase is produced by Moraxella catarrhalis? Simply because the medium, when it is uninoculated, has a pale green color because of the complex formed between DNA and methyl green. And if the organism present is Moraxella catarrhalis, its DNA cleaves DNA and methyl green complex. So it destroys the complex, and as a result, it would make the green color fade, and therefore the colonies will become surrounded by a clear zone, indicating that there is successful hydrolysis of DNA by the enzyme DNase. Another test, as I said earlier, is the butyrase esterase reaction using bromochloridolyl butyrate. And once butyrate esterase enzyme is produced by Moraxella catarrhalis, it cleaves bromochlorinodyl or bromochlorinodyl. I'm sorry. Once butyrate esterase is produced by Moraxella catarrhalis, it cleaves bromochlorinodyl butyrate into endoxyl and oxygen, resulting to the appearance of an indigo color. Again, the substrate is bromochlorinodyl butyrate. Sorry for the tongue twister. And in the presence of butyrate esterase, it can be cleaved to endoxyl and oxygen, resulting to an indigo color. Okay, here ends my discussion on the different Neisseria species, including pathogenic ones and non-pathogenic ones and related organisms. Please make sure to read all of these references. If you have any questions, please don't hesitate to ask your teachers. Here ends our discussion. Thank you very much, and God bless everyone.